Okay, the next speaker is Maximilian Press.
Hi, everyone. Thanks again for being here so early, and thanks to the organizers for letting me be here. So before I get too bogged down in the details, I wanted to start by talking a little bit about this paper by Francois Jacob from about 40 years ago. In this paper, Jacob makes certain arguments about how evolution works in biology. Namely, he says that evolution is not an engineer in that it does not come up with new biological plans de novo, but rather that it acts, in his words, more like a tinkerer, by which he means that it takes existing biological plans and fiddles with them to make them a little bit better or different. So I always thought this was a very interesting idea. I wanted to talk over the course of this talk mostly about one of the most important tools of the tinkerer, which is duct tape. So duct tape is an easy way to come up with quick fixes to immediate problems, even though that problem may not last too long in the long term. So in thinking about biology, I was curious. I thought a lot about what the biological analog of duct tape would be, and the best answer I've come up with, although I don't think it's perfect, is short tandem repeats. So if you don't know what these are, they are small nucleotide elements. So for instance, a unit of STR would be CAA, and this is 3 repeats of a CAA, STR or microsatellite. And these are quite common in regions of the genomes that you think of as being functional, which is a little bit surprising given that they have a rather high mutation rate. So you might expect that selection would try to purge them from functional regions. In contrast to this expectation, there's a bit of work that argues that there's actually active positive selection maintaining STRs in functional regions. In the Quantitative lab, we have a favorite STR, and it's the STR in the ELF3 gene of Arabidopsis thaliana. What I'm showing here, and it encodes a polyglutamine. And what I'm showing here are just 4 transgenic plants over here, which are expressing different alleles of that ELF3 repeat, which encodes a polyglutamine. And as you can see, there's a fair amount of phenotypic variation in terms of flowering time, which is a trait controlled by ELF3, such that some of them look basically like ELF3 null mutants and others look like wild types. As you'll notice, the endogenous repeat to this particular background, Columbia, is one that seems to repress flowering properly. However, this changes a bit if you look in a different genetic background, which is the WS genetic background. Again, the endogenous repeat is the best at complementing this phenotype. However, it is actually a different endogenous polyglutamine in that strain of Arabidopsis. So this is a pretty standard example of sign epistasis, with the only wrinkle being that there are actually a huge number of alleles of this particular microsatellite that are all naturally available in Arabidopsis. So there are a few mechanistic ideas for how this could be working in this particular instance of sign epistasis. And so what ELF3 does in the cell is it just sticks to other proteins in a combinatorial fashion. So our first hypothesis for what was going on here was that it was just in some polyglutamine variation was destabilizing protein interactions in some way, leading to this mutant phenotype. However, there is this problem that in a different background of Arabidopsis, you've got a different polyglutamine which seems to be working just fine. So our hypothesis for explaining this was that there was a corresponding polymorphism in the interacting protein, which I'm trying to show here, different shape. And following that, so our idea for how that works is that corresponding polymorphism in the interactor happened first, and that the ELF3 polyglutamine actually compensated for the first polymorphism. And this is what I really mean when I say that STRs could be like duct tape, is that they provide a high mutation rate for solving problems that might arise, such as destabilization of that interaction. So I'm going to provide a couple of lines of evidence that this could be the case, at least for the ELF3, polyglutamine, or rather the polyglutamine encoding STR, the first being a genetic mapping approach, and the second being actually looking at the protein-protein interactions of ELF3. It's a pretty standard genetic mapping approach. You take these two genetic backgrounds I've already discussed, the Columbia and WS. For the sake of argument, I'm going to say that ELF3, this sign epistasis is due to ELF3 interacting with some gene called gene 1. If you make the F1, this is a wild type looking plant, which makes sense because, at least according to our model, because there's still favorable interactions that can be made between ELF3 and gene 1 based on the matching of colors. However, if you follow to the F2 generation, you expect ELF3 and gene 1 to segregate away from each other at some rate, and you will end up with mutant-looking plants. In this approach, I'm going to be using a somewhat different phenotype, which is a little bit easier to score at bulk than flowering time. And that's hypocotyl length. So what I'm showing here is an ELF3 null mutant, and as you can see, it's got a dramatically elongated hypocotyl relative to wild type. And when I actually look in F2s, what I see is that there's a fair amount of phenotypic variation for this trait among Columbia by WS F2s. And I can quantify this, and what I observe is that for the two parental lines, you see that there is a quite narrow and quite distribution and quite similar distributions between the parents. In the null mutant in green, you can see that it's pretty dramatically elongated. And that for the F2s, you see that they mostly look like the parents, but they have a long upper tail of transgressive variation. So this is some argument that there is an incompatibility that's being revealed by the cross, and also that this could be due to ELF3 because the phenotype looks similar. And so we did what everyone does these days, and we took the F2s, and we chopped off the tails and sequenced those and did genetic mapping. So this first pass QTL analysis shows a very nice peak right over ELF3, but then a number of other peaks. And so but this isn't really answering our question. What we really wanted was to find QTLs that were interacting with ELF3, and presumably not all of these QTLs are interacting. So what we did was a very puristic analysis, which was to take our F2s and then mask the genotypes in QTL analysis of all the individuals that were not homozygous for each of either the Columbia or the WS alleles of, sorry, I explained that poorly, mask all the genotypes of the individuals that were not homozygous for the Columbia allele, and then we repeated that same analysis except masking everything that was not homozygous for the WS allele. And with the idea that we would find allele-specific loci if there is in fact sign epistasis going on. And when we did this, we were surprised because it turns out that actually all of these QTLs interact with ELF3, which was somewhat confusing. And so what I'm showing here is that all of these, in red, all of these QTLs only show up if you are homozygous for the Columbia allele of ELF3. So, yeah, that was surprising. And so we looked at this a little closer, and we found a candidate gene that we liked a little bit on chromosome 4. And this is called LSH9. It's not very well described. So to show that this worked outside of the context of the F2s, I obtained an LSH9 null mutant, made double mutants with it in ELF3, and we were able to show that, yeah, it does still look like there's some sign epistasis going on between the null mutants also. So LSH9 normally decreases hypocotyl length just a little bit. It's robust, but it's a very small effect. But when you put it in an ELF3 background, the double mutant actually increases hypocotyl length relative to ELF3. So it does look like there is some basis, genetic basis for the sign epistasis that we observe in transgenics according to ELF3 STR variation. And again, our hypothesis for how this is working is that there's an underlying protein-protein interaction that is going on here. So this leads me to the second part of this talk, where I actually look at some of these protein-protein interactions. How I'm going to do this is using yeast-2 hybrid. Probably most people sort of know how this works, but I'll just go over it really quickly. How it works is you express two proteins in a yeast cell, and in a system where if they interact with each other, then that will turn on expression of some marker gene. And then you can select for the expression of this marker gene in your yeast cells and based on whether the cells grow or not, you can have an idea of whether the proteins interact. So we first did a positive control to make sure this works using a very well-known interactor of ELF3 called Phi-B. So Phi-B binds nicely to ELF3. It does not express anything with an empty vector, so that's good. And also, Phi-B does not care about any what version of the polyglutamine you throw at it. It interacts with them all more or less equally. So this is great. Next question was, we've identified this candidate gene. Can we come up with, can we find that there is preference for this polyglutamine for LSH9? So we did all the cloning and came up with the experiment, and it turns out that actually there's no physical interaction between ELF3 and LSH9. So this is a little disappointing, although it's nice because it encourages modesty and humility. The take-home message is that we were kind of wrong about the underlying model for how this is working at the cellular, at the mechanistic level. We think we know a little bit better what's going on now, but I don't really have time to talk about it. But we thought, this is a little disappointing, but as long as you got the system working, let's see if we can look at some of the many physical interactors of ELF3 and come up with an idea of whether the polyglutamine can do anything to protein interactions. And so it can, in short, although it's not super strong. This is one example, which is the well-known ELF3 interactor ELF4. It's quite a bit weaker of an interaction, as you can see here. The yeast are not happy at all, but it does look like there's a little bit of preference between these different versions. We can quantify that using the somewhat more quantitative LAC-Z assay, and it does look like the 16q allele of ELF3 is preferred at least above, say, this 0q allele. So that was nice. That, I guess, takes me through the second part. So there's, first of all, we can map these ELF3 dependent genetic incompatibilities, which are presumably due to the STR variation based on the transgenics. And second, there is a little bit of evidence that polyglutamine variation can disrupt protein interactions, at least at the subtle level. However, we don't really have any evidence at this point that 2 is the mechanism for 1. So I haven't answered a number of questions, the first of them being that we actually have no idea what the polyglutamine variation is really doing to ELF3, the protein, being one part. I only told you about one of 4 interacting loci, so I titled this talk is ELF3 is the hub of a network of epistasis or something like that. Well, the rest of the network needs to be fleshed out a little bit better. And lastly, I sort of started with this somewhat tortured argument about duct tape and how STRs can serve as these compensatory mutators of some kind. And we still, maybe that's the case for ELF3. It certainly looks like that from the genetic data, but we don't really know that for any other STR. And this last question is the one that we're most interested in at this point. So we wanted to look at it in more detail. The issue here is that actually with Illumina data and whole genome shotgun, you get extremely poor genotyping of STR variation across really any system. So we sort of approached this problem previously in the lab and developed a technology which makes this a whole lot easier, though it doesn't completely solve the problem. And so what we're ultimately doing right now is we're generating high-quality STR variation data across, for pretty much genome-wide, across wide diversity of Arabidopsis strains. And what we really want to do with this data is to identify instances, like we think ELF3 is doing, of diversifying selection. And the reason why we think ELF3 is doing this is just the incredible diversity of alleles that we observe for this relatively short microsatellite. So with that, I'll thank a lot of people who contributed in one way or another, and I'll take any questions in 1 minute.
Can you say something about how you do that genotyping?
Yeah, so if you know molecular inversion probes, what you do is you add some probes and they actually capture the relevant loci, so it's a targeted assay, and then you pull that out and make libraries from the captured region and you sequence that. Usually at really, really high coverage with pretty long reads, and that works pretty well. Okay. Thank you.
Great. Excellent.